So far we have discussed about monosaccharides, disaccharides, trisaccharides, oligosaccharides and polysaccharides regarding their structure. Now we'll discuss about the functions of carbohydrates, biological functions of carbohydrates. So we'll be learning few examples of carbohydrates with their biological functions, their functions in the cell. Say for example monosaccharides. Monosaccharides are the intermediates or the end products of biochemical pathways which are existing in the cell. So they take part in several metabolic reactions, anabolism and catabolism. Say for example monosaccharide like glucose. It is an energy source. It is a carbon source. So therefore glucose is important in the cell in relation to source as a carbon, source as a energy. So glucose is used as a major source of carbon and energy by most of the living organisms. So glucose is preferred as a source of carbon and energy by most of the organisms. So glucose, it is required by eukaryotes, required by prokaryotes, and it is responsible for transferring the carbon into several biomolecules which exist in the cell. When glucose is degraded, the process is called as glycolysis. Glycolysis leads to generation of energy in the form of ATP, adenosine triphosphate. You can see in this picture glucose, it is entering into this metachromatic granule. So it undergoes glycolysis that leads to generation of energy. Further, the products enter into several pathways that includes citric acid cycle that also generates a lot of energy, and finally the compounds are degraded or the electrons then enter into electron transport chain that again leads to generation of a lot of energy in the form of ATP, and finally the compounds are degraded in the form of CO2 and H2O. So that is the importance of glucose, as it serves as a source of carbon and energy. Another example of carbohydrate, that is disaccharide, is sucrose. Sucrose is commonly found in plants. It is not synthesized in animals. This is a major intermediate product of photosynthesis in plants, and it is a form of carbohydrate which is transported into the plant tissues. So it is one of the transportation form of carbohydrates. Third example, a disaccharide, that is lactose. Lactose is a disaccharide sugar which is abundantly found in milk. It composed of glucose and galactose. So therefore lactose, it also serves as a source of carbon and energy, because whenever it is metabolized by cells, it is degraded into glucose and galactose. Then these glucose and galactose are degraded in order to release energy, and the carbon from this glucose and galactose is assimilated into the several cellular biomolecules. So this is how lactose is also very important source of carbon and energy. There are some examples of polysaccharides which acts as storage polysaccharides. Say for example starch. Starch is found in plant cell, and the energy and carbon is stored in the form of starch in plant cell. Therefore, we call it as reserve food material. So whenever the energy and carbon is required by the cell, and if the easily available carbohydrates are not available, in that case, the stored food material is utilized for cellular metabolism. The sources of starch are potatoes, seeds, corn, and several plant materials. Another example of storage polysaccharide is glycogen. Glycogen is a storage polysaccharide which is present in animal cells, in humans. It is mainly stored in liver cells in case of animals. It is a polysaccharide made up with glucose subunits. Glycogen has a branch structure, but it is structurally complex than starch. Another function of carbohydrate is structural polysaccharides. That means these carbohydrates are important in the formation of structural cellular components of the cells. Say for example, these structural polysaccharides are responsible for giving toughness or the strength to the cell. That means they are responsible for giving structural integrity to the cell. For example, cellulose. Cellulose is a structural polysaccharide. It is a principal component of plant cell wall. So integrity or the toughness of plant cell wall is because of one of the component, that is cellulose, and that is why it is called as structural polysaccharide. It is also present in cell wall of some algae, giving the strength to cell wall. Cellulose is made up with glucose subunits, which we have already studied the structure. Like the structure is like amylopectin. Another function, with the help of example of deoxyribose and ribose sugar. Deoxyribose and ribose sugar are pentose sugars. That means they have 5 carbons in their molecular structure. That's why they are called as pentose sugars. Towards right hand side we can see the structure of ribose. There are 5 carbons, 1, 2, 3, 4 and 5. We can see at fourth carbon is attached to the 5th one. See the conformation around each carbon atom. Around this first carbon atom it is OHH or HOS depending on the beta or alpha positions. At carbon number 2 it is HOH. There is a hydroxyl group. Whereas if you compare the structure with deoxyribose sugar, as the name indicates, deoxy, it indicates absence of oxygen at position number 2. So the positions are highlighted here. Here there is absence of oxygen, so it is CHH. In case of ribose it is HOH, and that is the reason why this is called as deoxyribose. Ribose sugar is a component of ribonucleic acid. Deoxyribose is a component of deoxyribonucleic acid, that is DNA. This is in case of RNA. So they are the building blocks of RNA and DNA. One more example of structurally complex polysaccharide, that is cellulose. Pure cotton, which is produced by plants, it is a pure form of cellulose which is in the form of fiber. So this fibrous polysaccharide, it is very important in order to synthesize different structural units such as papers. So this variety of cellulose, it is very important for the synthesis of materials such as papers. One more very important example of a structural component which is present in the bacterial cell wall, it is peptidoglycan. We can see towards right hand side the structure of peptidoglycan. Peptidoglycan is one of the structural components important in the cell wall of both gram positive and gram negative bacteria. However, the percentage of peptidoglycan in the cell wall of gram positive bacteria and gram negative bacteria is different. The percentage of peptidoglycan in the cell wall of gram positive bacteria is higher. The percentage of peptidoglycan in gram negative bacterial cell wall, it is less. Peptidoglycan, it is also called as murine. In case of Archaebacteria, it is called as pseudomurine because the composition of this peptidoglycan in archaebacterial cell wall is slightly different. So as the name indicates, peptido, that is peptides, glycan, carbohydrate. This means it is a conjugated biomolecule which is made up with amino acids and sugars, and that is the reason why it is called as peptidoglycan. In this structure we can see that peptidoglycan is composed of NAM, NAG, NAM, NAG, NAM, NAG. This means that these are the building blocks of peptidoglycan. This is one layer of peptidoglycan which is made up with alternative units of N-acetylmuramic acid. NAM stands for N-acetylmuramic acid. NAG, N-acetyl glucosamine. Both are amino sugars. So these amino sugars are linked to each other alternatively. It forms a straight chain of NAG and NAM. This is another straight chain of NAG and NAM. This is another straight chain of NAG and NAM. In case of structure of gram positive bacterial cell wall, there are hundreds of such layers. In case of gram negative bacterial cell wall, the layers number is very less. So the concentration of peptidoglycan is higher in cell wall of gram positive bacteria. Peptidoglycan principally consists of two amino sugars. As I mentioned, they are abbreviated as NAG and NAM. They are arranged alternatively to form a chain. Further, this N-acetylmuramic acid has a short chain of amino acid. That means a short chain of amino acid is attached to NAM, that is NAM. Here also we can see this is the NAM from the below layer which has a chain of amino acid, and then these two chains of amino acids are cross-linked. They are cross-linked through a chain of amino acids which is made up with 5 amino acids, and that's why it is called as pentapeptide chain. So clearly we can see that this is one layer of peptidoglycan, NAG and NAM. This is another layer of peptidoglycan, and these two are cross-linked through this pentapeptide chain. So we can see the composition of peptidoglycan. It is made up with NAG, NAM, short chain of amino acids, and a pentapeptide chain. This is how all these peptidoglycan layers are cross-linked to each other to form a tough structure of peptidoglycan like this.